morning. Here we are, Tuesday the 12th, and today we're going to start going into exactly how DNA turns into a trait. Now, we've already talked about how alleles will determine what trait an individual gets, your dominant or your recessive. Maybe there's some form of codominance or incomplete dominance. There's all sorts of different interactions there. But now I want to talk about what actually allows that segment of DNA. Yesterday, we practiced looking at the spelling of it and telling what would be opposite. So if we had ATTC, we know that the other side of it is TAAG, right? Chargaff's rule. Now, how does that spelling turn into hair color, skin? How does it turn into muscle structure? Whether or not you're lactose tolerant or lactose intolerant. All of those things are based on our DNA. And so there has to be a mechanism that will change the DNA, the chemical, into the structure that we have. And that's our goal today. Today, we will be practicing, I will be explaining, and with any luck, you will be learning how our DNA turns into what we are. And that's a really cool series of steps. And it's going to be kind of weird to think about this once we go over it, and then we can kind of take all the pieces of this that's happening microscopically inside our cells, and then it's going to somehow turn into this. But it does. And it works every time for every living thing. So let's get started. All right, so we are, of course, still in Schoology because we always will be. Unit 4, Genetics. Week 5, just like yesterday, our DNA week. And if you look here, yesterday we went over our main notes up here. I had you guys practice your DNA and your letters just to make sure that you knew Chargaff's rule and what links to what. And then I had 2 videos that helped to support and reinforce this idea. Today, we're going to be starting with our protein synthesis PowerPoint right here. And then here is the worksheet that you're going to work on today. And please don't jump right to the worksheet. No, not yet, because there's a little bit of clarification there. And then I've included 2 more videos. The first one, I am going to show in class. I'm not going to do it here because that would be a YouTube video violation. And yeah, so if you're watching this on YouTube, you're going to want to go and click this one to watch the process, maybe even a couple of times. The second one here from the Amoeba Sisters, this explains everything that we're doing today just in one of their videos. So I will not be showing this, but it's here as resource in case you need it. So let's get into protein synthesis. All right, once again, this protein synthesis is how DNA becomes traits. And we've talked about this previously. Remember, everything that you are is protein. All of the traits that you have are either protein or due to protein. We are a protein structure. That's why we eat other animals, to get protein, because they're made of protein too. So this is how DNA becomes our traits, because it needs to make protein. That's all synthesis means, is to make. So first, let's identify exactly how this works. All right, so when we take a chain of DNA, the big polymer, we separate it into 3 letter segments. Every 3 segments, every 3 letters is what we call one codon. Now, each codon is going to be responsible for a different amino acid. Once again, going back to our earlier unit, unit number 2, our organic molecules, you hopefully remember that amino acids are the monomers of protein. Protein are the polymers. So these are the building blocks that make a protein. These are the little Lego pieces that when they all click together make something bigger and more complex. So AGG will give us the first amino acid, ATA the second, CCA the third, CGA the fourth, right? But there's an issue. DNA can't leave the nucleus. Remember, the boss has got to stay in the office, which means it needs a message. It needs some way to communicate out. So, what DNA does is it makes RNA, namely messenger RNA. Big in the news right now because the COVID vaccine that's rolling out nationwide that people are signing up for and making appointments for, that's a messenger RNA vaccine. So, literally, that vaccine is in this process. It uses the exact same machinery that DNA does. So it makes RNA. RNA leaves the nucleus. Well, when you get that vaccine into your arm, it's already out of the nucleus. And now that RNA will work with a ribosome to make protein. Hmm. So this is what it looks like. The DNA is in purple here. Remember, DNA is too big. It is too complex. It's all rolled up. It's all coiled. It can't leave the nucleus. It can't fit out through these little openings. But RNA can. So we open up the DNA. We make an RNA copy. That RNA copy leaves, comes out, and it clicks through a ribosome. That's this red thing. When these two ribosomal units come together, that's one ribosome. And now, as it threads through and it moves every chunk, each section, three different letters just click, 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 click. It is coding for what amino acid needs to line up next. Other molecules in the cell will find that amino acid and bring it in place. And then one by one, click by click, it will put together that protein chain composed of all the amino acids. So let's see how this works because you will be expected to do this. The Regents asks it every year. One of our state labs, this is a major section on, it shows up every year in the Regents. Step one, first we have to turn our DNA into our RNA. Now, for the most part, it's real easy because T still turns into A, C still turns into G, G still turns into C. The only difference is that messenger RNA, for whatever reason, does not have thymine. Instead, it has a different base called uracil. So where there should be a T, there will be a U instead. That's the only real change. Now, if you look over here in the box 3 insert, here's our DNA chain. T, C, A, G, C, A, T, G, C. Now, the messenger RNA copy of that will be this pairing. So T will link to an A, C will link to a G, A will link to a U, et cetera, et cetera. And you can go down the whole list. Now, there's an easy shortcut for this. Do it just like normal. A to T, C to G, and vice versa, and then worry about it. Because when you do that, you'll come up with T's and A's and C's and G's and whatever. But that T, when you look at the chart, whatever chart you have to use to read it, there is no T. Instead, all the T's will be U's. So then you just go back and change your T's to U's. It's really pretty simple. So that's step one. Then step two, our messenger RNA is going to connect with a ribosome. Remember the little dots that sit all over the cell? Their job was to make protein. Here's how they do it. So we're bringing now an earlier unit here and an earlier unit here. We're bringing them all together in how this works. Now, once it connects, what's going to happen is the ribosome is going to bring in each of these amino acids. They're going to match up to the 3-letter codon that we have on the RNA. And so, whatever the spelling of those 3 letters, that will determine the next amino acid. And then we go to this chart. Now, you remember how I said the easiest way to do this is just to use A, T, C, and G. And then when you look at this chart, take a look across the top and down the left-hand side. You see A, there's a C, there's a G, but what's missing? T. And so you just go back and you go, that's right. T becomes U. So then you go back and you replace your T's with U's. Perfect. And what we see here is that depending on the spelling, we find all the different amino acids. That's what each of these little lowercase kind of words are. So pro, sur, tier, his, glin, asin, lis, asp, glue, whatever. All of those are the shortened versions of our amino acids. So if the spelling was ACC, you would come over to the left-hand side for your first letter, A. Then you would come to the top for your second letter, C. You would find where those two meet. That's this box right here. And ACC is right here. That will give us the amino acid thir. So you'd write down THR. If the spelling was CGA, we would start with C. We would come across the top to G, bring it down until the two of them meet right in this box. And CGA is right here. That will give you the amino acid ARG. All right, so let's actually practice it. Looking at this, I've given you the DNA, 4 different codons. Now, I want you to look at them. And what would be the matching RNA to this DNA? Well, A is going to link to what? T. Or U, if you remember. T will link to A. G will link to C. So U, A, C. And the next one, C will link to G. G will link to C. A will link once again to T, but there is no T, so that's a U. And you could continue this down for all of them, step by step. Now, we want to take these and we want to find out what amino acid is being produced by each one. So the first one is UAC. So we go to U, we come across top to A, we come down, it's in this box. UAC right there makes TYR. Then GCU. So we come all the way down here to G and U from the top. It'll be in this one. Oh, no, sorry, C. GCU is right here. That makes ALA. So far we have tier ALA. GCC is in the same box. It also makes ALA. So we have tier ALA ALA. And then UGA. U and G. A is stop. So that would be our sequence. It would be tier ALA ALA stop. That's one complete protein, 3 amino acids. That's a really small protein. Most of the time they're bigger. All right, so let's try something more complex. All right, so first, ACT is the DNA would turn into what? A links to T, C links to G, T links to A. But then when you look down here at the chart, once again, there is no T, so turn your T's into U's. So U, G, A. Next, G will link to C, C will link to G, and once again, A will link to T. But since there is no T, it'll become U. All right, so A links to U, T links to A, C links to G. And you can continue this for each one. Wow, that went out of order. You can continue that for all of them. All right, so now let's decode this for the amino acids. UGA, we start at U on the left side, come to G along the top. UGA is stop. Okay, so that might have been the end of a previous gene, right? That's where that protein stops. All right, so next, CGU. Start at C, up to G. CGU will be in this box then, and it's right there. CGU is ARG. Then UAG. So U and A, there's U, it's another stop. Okay, so that one we just want to produce one amino acid. Maybe that's going to be used by something else. All right, starting up again. GGU. So G at the bottom, G at the top, it'll be over here. GGU makes gly. Then CAU. Okay, C on the left, A on the top. CAU makes his. Then GGA. So G on the left, G on the top, brings us down to this box. And if you take a look, GGA makes gly again. And then finally, GGU. Well, here's a trick. We already know GGU. GGU is right here. We already identified it once, so we can just copy it over again. All right, so now we know that this section of DNA makes this series of amino acids. One arginine, and then a protein of gly, his, gly, gly. Who knows what that makes? Maybe it's something like an enzyme. Remember, enzymes are made out of proteins. And enzymes do things like break stuff down. So maybe this makes the enzyme to break down lactose, the sugar in milk. And if you have the DNA spelling like this, maybe you are lactose tolerant. You can break down dairy, you have no problem with it. But maybe if we were to change one of these spellings, let's say we change the CCA here at the end, and we change it to CTA. Well, CTA is going to be GAU. If we look down here, GAU makes ASP, not gly. All of a sudden, we've changed the amino acid. That means we've changed the order. We've changed the shape of the protein. Now, all of a sudden, with that DNA code, you're now lactose intolerant because now the enzyme doesn't fold right. The active site no longer fits the substrate of the lactose. And now you cannot digest dairy. And now, if you drink milk, eat ice cream, have cheese, something like that, now you get really uncomfortable because the process doesn't work right, because the enzyme isn't the right shape. Ladies and gentlemen, this is how we do protein synthesis. And your assignment today will be the protein synthesis worksheet. Now, for this, I am not asking you guys, there are 8 of these. I'm not asking people to do all of them. I'm asking you to do 2 of them. And there's a little additional start thing here, and that is it says use your codon chart to determine the amino acid sequence. Remember to read through the strand and only start on AUG and stop when it tells you to stop. So here's the example. The DNA here, there's a full chain, but notice they don't start doing the protein until they get the right codon, AUG, for start. And then when they get GAA for stop, that's it. Done. So as you guys go through here, TAC will produce the RNA AUG. That starts. Like right here, this first one won't start until this third set in. Now, if you're like, well, where's the chart? I have one sitting for you right down here at the bottom. So you work left-hand side for your first letter, top for your second letter, bring those two together, and then you can find it. Or you can come over here to the right-hand side where it's organized for your third letter. And if you bring the side, the top, and then that side in, you'll find exactly what you're looking for. This is our goal. I strongly encourage everyone to go back and watch this video if you are not in live class with me in order to make sure. It shows this whole thing happen. It talks about what's happening. If you need another refresher, the Amoeba Sisters video is very, very good. Ladies and gentlemen, I hope that this helps to clarify how we turn DNA into the proteins that make us up. Tomorrow, we're going to take a deeper dive. We're going to look at exactly what happens when that enzyme is not formed right. I talked about it here at the end. Tomorrow, we'll see it. Ladies and gentlemen, I hope that you have a great day. I hope that everyone's finding this time virtual not quite as stressful as we've had in the past. And as always, of course, I look forward to seeing you all in class.